first property of water is that it is transparent, and I think that would be the easiest one to give a description of. That means that light passes through it. Now, what would be the significance to life that light can pass through water? Yes, John?
Insignificance to life, does that mean human life?
It could be any kind of life. Things that are living. Could be humans, could be other organisms.
What if made us able to handle spiritual during our development?
So we're talking about being able to find food. So I'm going to put, we can find food. And that would be true for us and many, many other organisms.
It helps for the plants to grow underwater, so it helps the whole ecosystem.
So I'm going to write the word photosynthesis, indicating that aquatic plants, not just plants on land, can perform photosynthesis. Does anyone have any other significance to light examples that they'd like to share? That photosynthesis one would have been my example. That's the one that I usually use. In the next one then, cohesion. And yesterday I added the word adhesion because they kind of go together. Cohesion and adhesion are both properties of water. And they are both based on the fact that water is polar. That means it has a positive end and a negative end to the molecule. Because of that, because it has a positive side and a negative side, kind of like a magnet, that means that water is attracted to water, and water is attracted to other substances. Just so we're clear, cohesion is water attracted to water. Adhesion is water attracted to other substances. So because water is polar, it sticks to other water molecules and it sticks to other substances as well. Now, what would be the significance to life that's brought about because of cohesion or adhesion? So the fact that water sticks together, or the fact that water sticks to other substances? Go ahead, share your example.
It allows plants to move water.
So both of those things, not just cohesion, not just adhesion, would allow plants to move water. You may remember learning in Science 10 about how plants have two types of vascular tissues, the xylem and phloem. The xylem is the one that transports water. Because water is attracted to other substances, it sticks to the sides of the xylem and helps it not fall back down the plant. Additionally, as soon as one water molecule evaporates through the leaf of a plant, it pulls another water molecule up with it. So because of cohesion and adhesion, plants are able to move water even though they don't have muscles or joints or anything like that. Does anyone have a different or another example of cohesion or adhesion being significant to life? I'm going to give you the example there's a thing called the Jesus bug, Jesus lizard, something along those lines. But the idea with the bug specifically, and not the lizard has to do with it walking really fast. But the substance is called the organism is called the Jesus bug because it literally walks on water. And the insect is so small that the cohesive force between water molecules is enough to keep the insect supported on the surface of water. And so that would be an example where cohesion is helping life exist because it's helping this organism stay above the surface of the water. That is called that is what the little bubble on the top of the water is called, the meniscus. Meniscus doesn't necessarily form in a lake where these would be. A meniscus is moreover when you have water in a container and you can fill the container a little Does anybody else have any other examples of significance to life they'd like to share? Yes.
So that we can cycle water out of rivers for aquaculture.
Cycling water out of rivers, and which force would you say we're talking about there? Cohesion or adhesion? I guess cohesion would be the one that would work the most. Anytime water moves in a stream, water molecules follow other water molecules. So it's always really important that water is attracted to water. Now in the category solvent, I could repeat myself and talk about water being polar, because that's the same reason that water acts as a solvent. Now what is a solvent? A solvent is something that dissolves and perhaps transports substances. So because things can be dissolved in water and be transported, because water flows, this has significance to life. The reason that things can dissolve is usually because water is a polar molecule, and so it will help pull two other pieces apart. Can anyone think of an example that's significant to life where it's good that something could dissolve and be transported in water? Yes. Sorry? What do you be more specific? What is the living thing that is dependent on that? If salt could be an example, if we relate it to life.
Like for saltwater fish, and saltwater stuff.
So, let's talk about saltwater fish. There would be an example where something needed to be dissolved in water and it was significant to life. I'm going to try to jog your brains and ask you to think about the beautiful red water that circulates through your body. And maybe this will clue you into a significance to life.
I was going to say Hydrolysis.
Which living organism? I want us to really focus on a living organism here, life, living organism. So yes, water helps us create medicines, that's for sure. But I really want to focus on life, living things here. And so let's go back to this red water I just mentioned. What am I talking about? Yes, blood. You have a lot of water in your body. A lot of it is in your blood. So let's think about blood. What gets transported in your blood? Does anybody know? Nutrients. Let's be more specific. What kind of nutrients? Calcium. Sure. Calcium is a mineral. I wrote Ca2 positive because that's what's actually kicking around in your blood, and I prefer symbols to words. But there's calcium ions in your blood, that's a mineral. You would also have vitamins in your blood. You would have oxygen in your blood. Now, you would have all of these things that you need, but your blood also helps you get rid of things that you don't. So there is also waste in your blood. And that would be a perfect example of why water being a solvent is significant to life. It helps bring things that you need to cells in your body. It helps remove things that you don't and get rid of them. Now the last property of water that I talked about here was thermal, and the idea I was trying to get at is that water has a very high heat capacity. That means that it takes an awful lot of energy to make water change temperature. So water is like a sink. It stores heat that can be used by the environment around it. So because water stores heat, we could think about sort of our planet as a whole, or even an individual organism, how water helps them survive. Let's think about a human person or any other organism that has water inside of them. Why might the fact that water stores heat be useful to life? Yes.
Water in your body keep you warmer.
Water in your body keeps you warmer for longer. So we'll talk about maintaining temperature. The water in your body keeps you warmer longer. If you didn't have water, the temperature in your body would fluctuate much more than it does. Now, if we talk about on sort of the scale of the whole entire planet, the fact that water stores heat could be very significant to life because water moves. So you might remember learning that things kind of start at the equator and then move away from it. Water can help transfer heat. And that would be the other example I like to use. So most of the sun's energy is absorbed at places near the equator where the sun is directly hitting without an angle whatever surface is there. And that heat doesn't all necessarily need to be used right at that spot. And so water can help transfer heat that is absorbed at the equator to other more polar regions of the planet, thus permitting life to exist. Does anyone have any questions about those four properties or the significance to life that they may have? So I'm going to pause talking here. All right, so I'm going to talk about these two processes now. So anytime I have stuff that's on the board, if I'm taking the time to show it to you, you should be writing something down. Now, it is a skill to be able to read something and make your own summarized version of it, and I encourage you to do that. So write something that you are going to understand later. If you prefer to write bullets or to make little diagrams, do that. This is just the information that I want you to know. So we're talking about macromolecules, and since chapter 6 is all about digestion, that's the focus that we have here. But really, any time these molecules are being broken down, this would be happening. So during digestion, we are going to break chemical bonds. That would be a key point as to what is happening in this process. There are bonds that hold the pieces of a carbohydrate, of a protein, of a fat together. So remember, they're macromolecules. They have lots of pieces that are all put together, and there are bonds that are holding that together. When I break those chemical bonds, it is called hydrolysis. If you look at the word, there are two pieces, hydro and lysis. Hydro means water, lysis means break. So literally we're saying water breaks the molecule. Now, the way that this actually happens is that I'm going to add, and I don't just mean I'm adding it, I'm chucking it into the container. I literally mean I'm going to add a water molecule to the molecule I'm trying to break apart. I add a water molecule right into the structure of the carbohydrate or the protein or the fat. And by adding that water molecule, the hydrogen and the oxygen in, I split the compound into two smaller compounds. So I have a picture here. It's very similar to the picture that is in your textbook. I would not expect that you could, from memory, draw complex molecules of sugar like this. That's not the point. I'm going to highlight one piece of the puzzle, and that is the piece that I want you to be aware of. These are two molecules of sugar. I'm showing you two opposing processes happening in the same picture. At the top, I have two individual sugar molecules. Here I have one larger sugar. And we'll talk about the names for all of these things afterwards. The process of hydrolysis happens when I go from the bottom to the top. When I look at hydrolysis, here's what I want you to focus on. In my picture and the picture that's in your textbook, we're looking for a spot where there's an oxygen kind of kicking around all by itself. It's usually an oxygen. It doesn't have to be, but that's very often the thing that we will find. And we're going to add a molecule of water. And I like to write water like this. H and then an OH. I'm trying to show that there's two sides to the water molecule, because this is how the water molecule is going to incorporate itself. So I'm still just talking about H2O, but I lined it up now. I wrote it the way the water molecule actually looks in real life. A hydrogen on one side, then an oxygen, then another hydrogen. This H and OH are going to literally insert themselves into the molecule. So they're going to be like little parasitic invaders, and they're going to go in that spot in the middle and attach themselves. Now remember I said there was this O all by itself. So with a bit of color, I'll show you. This hydrogen ends up here. I used to have an O all by itself. Now I have an O with the hydrogen attached to it. This OH is right here. There used to be nothing there, and I tacked it on. So I literally took the atoms from a molecule of water and put them in another molecule. And where they went in, that's where the molecule splits apart. So, do I care about this whole rest of what the molecule looks like? I do not. I care that you understand that at a point in the molecule, an H and an O and an H will get added in. And that is hydrolysis. Does anybody have any questions about that? So let's do dehydration synthesis then. Dehydration synthesis is the opposite process. That is when I build a large molecule from two smaller ones. And the way that I do that is exactly the opposite of what just happened. I will remove a hydrogen and I will remove an OH group from either a carbohydrate or a protein or a fat subunit, and that's what will allow them to join together and make a longer chain or a bigger molecule. It is called dehydration because I'm taking away water, and synthesis is a word we often use when we are producing something. So I am producing something by removing water. Now this reaction actually is a type of polymerization reaction that when we do Chem 30, we still talk about. So if you plan on taking Chem 30, when you get to the organic chemistry unit on polymerization, you actually already know this. This is exactly how it happens. Both of these processes, hydrolysis and dehydration synthesis, require enzymes. They don't just happen instantly as soon as water touches things. Other molecules are necessary to speed up the reaction and make it happen. And that's good. That's like a little safeguard so that anytime water touches protein, it doesn't break it apart. That would mean that anytime water touched your muscles, they would be broken, and that would be key. Does anyone have any questions about hydrolysis or dehydration synthesis before we move on? So I'm going to pause again here. So what is a carbohydrate? Well, its purpose, its function is to store energy. That is its main #1 function. If I asked you, what is the purpose of a carbohydrate, this is what you should tell me. Now I'm letting you know that it's also a structural compound. By structural, I mean it's part of the structure that holds things together. So for example, in cell membranes, there are pieces of carbohydrate as part of the structure. That's not its main function. Its main function is to store energy, but it does do other things. What is it made up of? It is made up of carbon, hydrogen, oxygen. I would like to highlight that it's made up out of carbon, making it an organic molecule as opposed to something that is not made with carbon. That would be an inorganic molecule. So it's made of carbon, hydrogen, oxygen. Now when we're talking about carbohydrates, and we want to talk about dehydration synthesis and hydrolysis, we need to be aware that there are different types of carbohydrates. Another word we could use for carbohydrates is sugars. So a sugar is a carbohydrate. A carbohydrate is a sugar. They're sort of interchangeable. We have a few different kinds of sugars that exist. We have mono, which means one, saccharides, which is a big long word that means sugar. Monosaccharides all have something like C6H12O6 as their formula. So they have something like 6 carbons. I wrote down that particular formula because it's the formula for glucose, and glucose is the one carbohydrate whose formula you actually will need to know. We're going to use it in photosynthesis and cell restoration at length. So you need to be aware that that is glucose. Now there are other monosaccharides. Glucose, fructose, and galactose are all examples. And I'd like for you to notice something similar. They all have OSE as part of their names. Glucose, fructose, galactose. Now these are examples so that I can use them to describe the next type of sugar. A monosaccharide, for some reason my picture's a little faint over here, so I'll just re-sketch it. A monosaccharide usually looks like a really poorly drawn hexagon. I don't draw things, this is why, this is how hexagons come out. But a monosaccharide will have one of these units in it. It's sort of the basic, smallest piece of a carbohydrate that you can have. Now, if mono means one, then di means two. I can also have disaccharides. Disaccharides usually have a formula that's very similar to C12H22O11, which is sucrose. Sucrose is sugar. Like if you think sugar, like what you eat, that's sucrose. So that would be the basic sort of size comparison. If you look, it's about twice as big. It's not an exact double because of the way the bonds are arranged, but if you look at the number of carbons, it goes from 6 up to 12. So a disaccharide would be something that has 2 hexagons attached together. Now there are 3 disaccharides that I would mention. Sucrose is one of them. Maltose is another. Lactose is the third. Notice that just like the monosaccharides, they all end with OSE. Now the last category are polysaccharides. Poly means many. So a polysaccharide is something that is made up of many subunits of sugar. Now some really good examples of polysaccharides are glycogen and starch, which are both used for storage. A polysaccharide, in either case, would look like many, I'm drawing 4, but it would realistically be many more than that, many pieces of sugar attached together. They could be in a chain, there could be branches coming off of the chain. It doesn't have to be in one straight line. The key would be that there's lots and lots of subunits. If you have something better to say than I do, then please come up here, wear my headset, and teach biology. But if you don't, can you please keep it to yourself so that the people that are next to you can hear me as opposed to you. Now on the side, I put a picture that had actual sugars in it, not because I want you to memorize it, but so that I could show you that sugars undergo dehydration synthesis from monosaccharides to disaccharides. So in my actual example here, I have glucose and I have fructose. Now, do I care that you memorize what the shape of these molecules is? I do not. What I care that you understand is that glucose and fructose are small. They are monosaccharides. If I take them and I put them together by the process of dehydration synthesis, like we saw before, so I take an H and an OH and I remove it off to the side, I can make one bigger molecule called sucrose. And this spot in the middle is where that water molecule used to be. So again, I don't expect you to memorize complex structures. I just want you to know what dehydration synthesis is. And specifically for a carbohydrate, that two monosaccharides could make one disaccharide. And to help you keep that straight, here is a list. How do I make sucrose? How do I make lactose? And how do I make maltose? Sucrose is made up out of glucose and fructose. And I'll tell you a cheesy little way I remember this. Sucrose to me is sugar. That's what sugar is sucrose. And when you eat sugar, you get all hyper. So I say to myself, sugar, S, makes you go, G, fast, fructose. It's lame, I realize that, but sometimes the things that are the silliest are the things that you remember. Now lactose is what's in milk. It's often what people have trouble digesting. We can talk about that later in the digestion unit. Lactose is made from glucose. And notice that all of them are made out of glucose. So another trick that I have is to not remember glucose. Everything has glucose. But to try to find a trick to remember the second monosaccharide. In lactose, the other monosaccharide is galactose. It has the word in it. And so I find that one typically easy to remember because it has the word lactose in it. The last one is maltose. It's made out of glucose and glucose. So not two different subunits, but two of the same subunit. And so those are the disaccharides and the way that I would make them out of monosaccharide sugars. Would anybody like to ask any questions about the purpose, the types of polymonodisaccharides that are up there? I will just mention, I realize I didn't right here, glycogen is what is found in your muscles. Starch is typically what would be found in a plant. So that's an example of a polysaccharide in an animal or in a plant cell. Now the other topic I wanted you to look at today were fats, and so I will switch to that screen. Is there anybody who is still writing something down and would like me to move this one up? Good, are you good over there? See you writing something, but you're okay? Okay. So we are going to talk about lipids, which is the fancy name for fats and oils. Just as an interesting note, this is not something I would ask you ever, but fats and oils store 2.25 times more energy per gram than any other biological molecule. That's why fat is such an efficient thing to have. The purpose of a fat is to store energy, and you'll say, that was the purpose of the last one.
There's one fat molecule, all those high triglycerides.
Yes, we will get there, but that is one triglyceride fat. So the purpose of a fat is to store energy, and that is the purpose of a carbohydrate. The difference between a fat and a carbohydrate is that carbohydrates are really readily accessible, therefore immediate use, Fat is more for long-term storage, and it has a bunch of other functions as well. It protects your internal organs. So it's actually pretty important to have some fat as a cushion around the organs inside your body to help them protect them from shocks. Now, too much fat is bad for them, but a little bit is good. Fat is necessary to make hormones. There are some hormones that require fat, and it is absolutely a structural component of cells. So fat is not the devil. Fat is good. You require it. The problem with fat is that it can become too numerous very quickly. Now you'll notice that the title of this was lipid, and then I split it up into fats and oils. Typically, when I say a fat, we're talking about solid molecules that are produced by animals. Typically, when we talk about an oil, we are talking about something produced by plants. It could also be produced by animals, though, but it's generally a liquid. Now, this is not an all-encompassing 100% of the time definition, but this is usually what we mean. If we say fat, we're talking about something solid that an animal has in its body. If we say oil, it could be a plant or an animal that's producing it, and it's generally a liquid. Now, we're going to focus on one kind of fat. It is not the only kind of fat in the universe, but it's the kind I would like for you to know about. We're going to talk about triglyceride fats. A triglyceride fat, like its name would suggest, is made out of 3 pieces of fat and some glycerol. So we will have 1 glycerol molecule and 3 fatty acids. And so much like your textbook illustrates to you, I'm illustrating here a very similar looking picture. I don't expect you to memorize all of the details of the picture, but I'm going to pick out some of the distinguishing characteristics that would be important that you know. The first thing that it's important that you know about is that the glycerol is like the backbone. And in fact, a lot of the time when fats are discussed, people would say there's a glycerol backbone with 3 fatty acid chains on the side. That means that the glycerol in this picture is that chunk. Up here, it's this piece. Glycerol is a molecule that has 3 carbons on it. And it'll always be on the side of the molecule. So that's how you'll be able to identify the glycerol. The fatty acids are always the chains. So these bits down here are the fatty acids. Each one of these is a fatty acid. Now, why is it called a fatty acid? Because it's an acid and it's used to make fats, so it's fatty acid. It's not a particularly large acid, it's just the kind of acid that is used to make fats. Now, what I'd like for everyone to be aware of, just like for carbohydrates, is that for me to make a triglyceride fat, I need to undergo dehydration synthesis. So just like the other types of macromolecules, for me to put together glycerol and 3 fatty acids, I need to take out a hydrogen, an oxygen, and a hydrogen anytime I want to make an attachment. Notice how there are 3 droplets of water? That's because this has to happen 3 times. There are 3 chains that are going to get attached. So I'm going to have dehydration synthesis happen 3 times. Would anybody like to ask any questions about the structure or about dehydration synthesis? So then on the bottom, I have some categories of fats, just so that you can help distinguish different things that are going on. When we're talking about fatty acids, the things that will make up the chains in the structure of our triglyceride fat, I can have poly, monounsaturated fatty acids. A polyunsaturated fatty acid, where poly means many, just like it did with polysaccharide, is something that has many double bonds between particles. A monounsaturated fatty acid has one double bond and is illustrated at the bottom. If you look at those three fatty acids, two of them are a long, straight line. And the third one has a little kink in it, a little bend in it. And the reason that it does is that right here, instead of those two carbons being attached by one single bond, they're attached by two bonds. It makes a little bend in the chain. The word saturated is used because in a saturated fatty acid, which would be either of the top two, there are no double bonds. In a saturated fatty acid, if I were to count, and I wouldn't expect you to, but if I were to count how many hydrogens there were, on the top two, there are more hydrogens than there are on the bottom two. That's what the word saturated is talking about. Do I have as many hydrogens as I could? If I do, it's a saturated fatty acid. Do I have fewer hydrogens than I could? If my answer is yes, then it's unsaturated. So that third chain, if we were to count, has up 2 less hydrogens because it has a double bond instead. Would anybody like to ask any questions about what polyunsaturated, monounsaturated, or saturated means when we're talking about fats? So it would be realistic for me to ask you with a picture, hey, is there a saturated fatty acid around here? Is there an unsaturated fatty acid? And because each fat molecule would have 3 chains, you should be able to compare, is it straight or bent? Or if you needed to, does it look like one of them has less hydrogens around it than the other? Now the last thing that's on here is cholesterol, and I'm actually not going to say anything about it, so I apologize if you already wrote it down. Just pretend it wasn't